Question 25 will be divided into two parts of the video because it will take much longer time to explain these questions. So the part one will be explaining the question 25A. 25A, it will be in the part one video solution. While for the part two, it will be explaining the question B and C. So for this part one, as you can see in the questions, it is stating three types of the gene in the maize plant. So first one is the color of the seed, where purple, the purple seed would be the dominant one. Purple would be dominant, while yellow would be recessive. That is the first, type of character. And the second is either the seed is smooth or shrunken. If let's say it's a smooth one, then it's dominant. If it is shrunken, then it's recessive. And the third gene will be whether the seed is starchy or it is sweet. If let's say it's starchy, it is dominant. If let's say it's sweet, it is recessive. Right? So there will be, three types of the gene here. Okay. So now the first question you look at that, it is asking us to state those genes that are genetically linked. Genetically linked, So obviously this question is related to the link gene. So if let's say it's a link gene, then that means that it is related to the deviation. It's related to the deviation of the Mendel's model, and then it is related to the link gene. Okay, so for link gene, when we solve the questions, when we do the calculation, please remember we need to use the method type 2. The method type 2. Okay, so now let's see. State the gene that are genetically linked. So in order to determine whether the gene are linked or not linked, well, there's only one way. That is, we have to find the recombination frequency. Okay, you have to find the recombination frequency between the gene. Okay, so now the first condition or the first story that is given is this one. Okay, a heterozygous plant with purple and smooth seed will cross with homozygous plant with yellow and shrunken. Okay, so purple and smooth. Purple and smooth. So as you can see here, purple is dominant. Smooth is also dominant. Okay, so that means that purple and smooth, this one is actually big P, big S. At least one big P, at least one big S. Okay, they are crossed with the homozygous plant with yellow shrunken. Yellow shrunken, both are recessive. So yellow is actually a small p, and then the shrunken one is actually the small s. Okay, so in other words, this is actually, yes, a test cross. Yes, this is actually a test cross. Okay, so after these two individual is crossed and they produce this number of the offspring. They produce this number of the offspring. Okay, so you have the number of the offspring. So actually, you can try to figure out what is the recombinant frequency. Okay, so this is what we will do for the first one. For this P gene and S gene, So if let's say after we calculate the recombination frequency for this P gene and S gene, if let's say it's lesser than 50%, then that means that the P gene and the S gene, they are linked. If let's say it's more than 50%, then that means the P gene and S gene are not linked genetically. Okay. So now for this individual, it is heterozygous, and this is link gene, right? So for link gene, well, we will try to write like this. Either for heterozygous, right? Either you will write as P, big S, big P, big S, then small p, small s, because the question already state that that is heterozygous. All are heterozygous. Okay, so we can state like this, or we can also state like one big P and then one small s, then slash, then one small p, one big S. Both also heterozygous, right? Both also give this type of phenotype. So which one we should follow? Which one we should follow? Okay, so since we assume it is link gene ID, so we are using the type 2 method. Type 2 method is still the same thing. You have to find the genotype of the parent, then find the gamete, and then find which one is parental, which one is recombinant, then only you do the cross. Okay, so that is the type 2, right? Slightly different from type 1 method, yeah? Okay, so back to this, which one we should choose? Yeah, which one we should choose? Okay, so the tips to choose this is basically we look at the phenotype of the offspring. We look at the phenotype of the offspring. Okay, so if you see that the offspring is having those both dominant trait and both recessive trait, those offspring, if let's say they have higher number, then you should choose the one with 2 dominant together and 2 recessive together. Okay, but if let's say you find out that those offspring that is having 1 dominant, 1 recessive, 1 recessive, 1 dominant, if you find this type of offspring is having higher number, if let's say this is 200 something, higher number, then we will choose this one. Yeah, we will choose this one. So it depends. Yeah, this is the hint to help you to choose. Okay, so as you can see here, the one with both dominant, the one with both recessive have higher number in the offspring. So that's why we will choose this one as the genotype of the parent. Okay, we will choose this one as the genotype of parent. So this one we will not choose. Okay, so now we have the parental genotype ready. So now we will write it down. So this is for question A. Yeah. All right, so we check on the first pair of the gene first, the P gene and the S gene. So for parent, okay, so it would be heterozygous, is this one. Okay, big P, big S slash small p, small s. So big P, big S slash small p, small s. Okay, then it is a test cross. So we will cross with this one. Yeah, we will cross with this one. So you're right in the way of the link gene as well. So small p, small s, small p, small s. Okay, so done with this, then next we will write all the gamete down. Okay, so this will need us to have the Punnett square. All right, so the first individual, this individual, so same thing. Normally, this is what I will do. I will draw 2 lines. Right, I will draw 2 lines to indicate the chromosome. So big P, big S, I will write down, small p, small s. It's on the same chromosome, right? So that means that automatically big P, big S is one parental, small p, small s is another parental. So I will write down these 2 parental type of the gamete. Then I will draw the line. Then after that, crossing over should happen to produce the recombinant. So it's a big P, small s. That's the recombinant type, yeah, due to crossing over. Big P, small s. Then another one crossing over. So it's a small p, big S. Okay, so these are the parental type. These are the recombinant type. Then for this individual, is only going to produce one type of the gamete, so which is small p, small s. Okay, so we will do the cross then. Yeah, we will do the cross. So that will be big P, big S slash small p, small s. Then small p, small s slash small p, small s. Then big P, small s slash small p, small s. Okay, so done with this, then of course, what we can do is we will look into the phenotype. Yeah, look into the phenotype here. So based on the phenotype here, the first one is big P, big S, yeah, then small p, small s. So this is actually the one with the purple smooth. Yeah, big P, big S is purple smooth, right? So purple smooth, where it is, this is the purple smooth seed. So purple smooth seed number is 103. So you will write that down, 103. So 103. Okay, then next one, this one is small p, small p, small s, yeah? So this is homozygous recessive. So this one is basically the phenotype should be yellow color and shrunken, yellow shrunken. So yellow shrunken is here. Yeah, yellow shrunken is here. So total number is 105. So you will write down 105. Okay, then next one, this one would be one big P is a purple. purple shrunken. Yeah, purple shrunken is here. So total number is 28. So total number 28. And then lastly is the yellow, small p is yellow, yeah. And then this is smooth. So yellow smooth, yeah. So yellow smooth is 30. Yellow smooth is 30. So now you know that this is recombinant, this is parental. Okay, so can we get the recombinant frequency? Of course, yes. So the recombinant frequency, right, it will be equals to, so the recombinant will be 28 plus 30. Okay, then you have to divide with the total. Yeah, the total. So the total is 103 plus 105 plus 28 plus 30. So if you sum up everything, okay, so and then don't forget to times 100%. And this is what we'll get. This would be 58. Then this one would be 266. Yeah, so 266. So you times 100%. So total we get is 21.8%. Yeah, 21.8%. So if it is 21.8%, then is it linked? Of course, yes, it is linked. Okay, so that's why, yeah, P and S gene are linked. Yeah, P and S gene are linked. Okay, so on the other hand, how about the P and M? Right, because over here it stated the P and M, yeah. So color and also starchy or sweet, yeah. So it's between P and M gene. Okay, so this is another set of the test cross here. Okay, so same thing. In order to determine whether P gene and M gene are linked or not linked, all right? So yeah, we have to do the same thing again. Yeah, we have to do the same thing again. Yeah. Okay. So that's why again, first thing you have to find the parent. Parent is a heterozygous purple starchy seed. Yeah, purple starchy. Okay, purple starchy. Yeah. So purple starchy and some more is heterozygous. Okay, so purple is basically big P. Okay, and then starchy is basically big M. All right. Then test cross was will test cross, right? So test cross will means that they are going to cross with small p, small m, small m. All right. So done with this, then yep, we have to find the genotype of the parent. Yeah. So again, this is heterozygous P and M. So it can be, again, if let's say it's a link gene, it can be a big P, big M slash small p, small m. Or it can be big P, small m slash small p, big M. All right, it can be either one. Yeah, it can be either one. So how to choose? Which one we should choose? Again, it is the same thing. We look for those that is purple, yeah, those that is dominant, both dominant, purple and starchy. And then both recessive, yellow and sweet. Yeah. So purple and starchy, yellow and sweet. Okay. Purple, starchy, yellow, sweet. You can see that the number. Okay. So these two number is around around 100, 200. Yeah, 200. Okay. So this one is slightly higher compared to the compared to the purple and starchy, yellow and sweet. Yeah. So these two is higher. Right? The one with contain one dominant trait, one recessive trait one, right? You can see here, starchy is dominant. Okay, so these two is higher. So if these two is higher, then yep, we will choose this one. Okay, we will choose this one. Okay, because these two are higher. Okay, so if like this, then yep, we will choose this one then. So we have confirmed the genotype of the parent. So now we will do the same thing. Yeah, we will do the same thing. So parent. Okay, so parent would be, the first one is P small m slash small p big M. Okay, then second, we'll do the cross. Okay, so it's a test cross. Yeah, it's a test cross. So it's again small p small s slash small p small s. Okay, so after that, then we will do the cross. Okay, so based on the cross, again, I will draw two lines to indicate. So big P, small p, big M. Okay, so the parental would be big P, small m, and then small p, big M. Then we draw a line. Okay, so these two would be the parental. Then next, we will do the crossover. So big P, big M, big P, big M. Okay, then small p, small m, small p, small m. Okay, so these two would be the recombinant. Okay, then next, how about this individual? This individual is going to only produce one type of the gamete. So that is small p, small s. So once we have done with this, then yep, we will do the cross. Hey, sorry, how come that's an S? Okay, careless mistake. Okay, so this one should be small p, small m, small m. Okay, so this would be small p, small m also. Okay, once we have done that, so now we will look at the number. Yeah, we will look at the number. Okay, so this one is one big P. Yeah, one big P. Yeah, so one big P and two small m. Yeah, so one big P and two small m, it will be purple and sweet. Yeah, so purple and sweet. So purple and sweet is 98. Yeah, purple and sweet is 98. Okay, so purple and sweet. So second one is contain one big M, but two small p. So two small p would be yellow. Okay, and one big M would be starchy. Yeah, so it's yellow starchy. Yellow starchy. So yellow starchy, total number is 105. Yes, 105. Okay, then next one, big P. Okay, big P, big M. One big P, one big M is purple starchy. Yeah, so purple starchy. So it was 101. And then lastly is yellow sweet. Yeah, yellow sweet is 99. Yeah, yellow sweet is 99. Okay, so with that, then now we will try to do the recombinant frequency. Okay, so recombinant frequency equals to 101 plus, this one is 99, sorry. Okay, plus 99. Okay, then we will divide with the total number. I will divide with the total number. So total number here is we take 98 plus 105, okay, plus 101 plus 99. So total number. Then don't forget to times the 100%. So 101 plus 99 is 200. So divide by 403. Okay, so you times 100%. Times 100%. Okay. So this, let me get my calculator. Okay, so 200 divided by 403. So what we get is, it is 49.6. Yeah, 49.6. Yeah, so it's 49.6%. Okay, so it's almost 50. Yeah, it's almost 50. Yeah, okay. So if it's almost 50, then that means what? That means that, yeah, this most likely is not a linkage. Okay, reason is because it is too close already. Yeah, it's too close already. It's just different by just one, or different by just I think the recombinant and the parental. Yeah, so parental total is, sorry, recombinant total is 200. The parental total is just 203. So it's just different by 3. Okay, so that means that, yeah, it is very close. 49.6, you round it off, is like 50%. That's like 50%. Okay, if we change it to another way, that means that if, let's say, this is recombinant and this is parental, then the percentage is also around 50. right? If you count that, it's around like 50.4%. Okay, so 50.4%, it's the same. Yeah, it's almost the same. It's 50%. So in that case, then that means what? That means that this one most likely is not genetically linked. Yeah, most likely they are not genetically linked. Okay, so because it's too close. If let's say it's like 49.0, then yes, still can consider that. But then this is too close to 50. Yeah, it's too close to 50. So that's why, yeah, we will say this is not the linked gene. Okay, so if it's not the linked gene, then that will mean that those that's genetically linked, yeah, those that's genetically linked is only, yeah, so hence the P gene, only the P gene and the S gene are genetically linked. Okay, so these P gene and M gene, maybe they are on the same chromosome, but they are too far away. Yeah, they are too far away. So that's why they are giving this. Yeah, they are giving this number. It's like very close to 50% already. Okay, so it is almost same like the normal Mendel's model already. Yeah, if let's say you do a test cross, it will give you almost like same thing, 1 to 1 to 1 to 1. Okay, you take 105 divided by the smallest number, it will just still give you 1 to 1 to 1 to 1 ratio. Okay, so because of this, that's why, yeah, it is too close to each other. So even though maybe the P gene and the M gene, they are occurring on the same chromosome, but then they are too far away. So in this case, they are not genetically linked. Yeah, they are not genetically linked. So this is the example where the gene are physically linked. They are on the same chromosome, but then genetically they are not linked because they are too far away. Yeah, because they are too far away. Okay, so since the question only want the gene that is genetically linked, so that's why we can only list the P gene and S gene, not for the P gene and M gene. No, only P and S. Yeah, only P and S. Okay, because they are asking for those that's genetically linked. All right, so that is for the question A. So for the question B and C, we will discuss that into in the part B. Yeah, in the part B.